I'll introduce you. Hello, everyone. Welcome to the MIF++ seminar. It's a pleasure to introduce Professor Tom Grant from the University of Buffalo, Department of Structural Biology. Tom will talk about information-theoretic approaches to the inverse problem in solution scattering. So over to you, Tom, please.
Great. Thank you so much. So thanks, first of all, Vitaly, for inviting me out. This is a great opportunity. I often don't get a lot of opportunities to talk about a lot of math, so this is really great for me. I'm a structural biologist, but I'm more on the sort of physics and math side, developing computational tools for structural biology. So this is a great opportunity for me to talk to a group that likes math and likes hearing about it. So we'll go through some of these approaches that we've developed for applying information theory to the problem of solution scattering. So I'm not much into material science myself. I don't have a lot of experience with it. So I'm not sure how many people here are familiar with solution scattering and with structural biology, but looking through some of the talks that previously have been in this series, I saw some people have talked about structural biology. So I thought I'd give a quick overview of the basic experiment of solution scattering before we get into it. So on the upper left, we have a protein molecule represented as an electron density map. So this is a 3D volumetric grid where every point in real space has a value, a density for the number of electrons at that location. And if we can calculate the Fourier transform of this 3D electron density function, we get what we call the molecular transform. And the molecular transform is a complex valued mathematical function that has amplitudes and phases. Okay, every point in this reciprocal space has an amplitude and a phase. And if we knew every one of these amplitudes and phases, we could calculate an inverse Fourier transform and get back at the electron density function of the particle. And that tells us a lot of information about the structure of the particle, and we can do things like design drugs and biology with that information. So it's very useful practically for us to get information about the structure of proteins and RNA and other biological macromolecules. Now, the molecular transform is sampled in a way through X-ray scattering experiments. So on the bottom here, we have what you would see if you could actually measure a single molecule X-ray diffraction image. You can see that I'm rotating the molecular transform here, and that's because as we rotate a molecule in 3D, we're also rotating the corresponding molecular transform as well. And so we get a cross-section through this molecular transform depending on what the orientation of the molecule is. But of course, we can't do an actual single molecule diffraction experiment in practice. There's simply just not enough photons scattered on the detector. There are techniques involved currently being developed to try and do this with tools such as X-ray free electron lasers, which is a field I'm involved in. But it's not something that's routine. It's very hard to do. And it's one of these things that seems to always be 10 years down the line kind of a thing. So what people can do is solution X-ray scattering. So we can have these molecules freely floating in solution, but rather than a single molecule, we have billions of them, right? Just a simple dilute solution of molecules. Maybe 10 mg per mil is a typical concentration of protein that's in a small vial. And these molecules are freely floating in solution, and because they're tumbling, their scattering patterns are all from different orientations of the molecule. So when you take all of those individual single molecule diffraction patterns and you add them all up, you end up with this sort of isotropic, smeared out scattering pattern on a 2D detector. Okay, so you get this bright blob of intensity at the middle of the detector, and then as you go to higher angles on the detector, you get weaker and weaker scattering. So because of this spherical averaging, we lose a lot of 3D information in these experiments. But the advantage of these experiments is that they don't have a lot of the constraints that other structural biology experiments have. Typically, we use things like X-ray crystallography or cryo-electron microscopy. And so that means the molecules are in a crystalline state or they're frozen, and that means they're not really close to their native solution conditions, right? And so solution scattering allows us to probe low resolution information about the overall size and shape of the particle in solution and at room temperature. And that's its major advantage. It's a very complementary technique to these other techniques. So you notice that we have this bright intensity at the middle, and so a lot of times we collect data at small angles because that's where we get a lot of photons, a lot of signal to noise. And so we often call this technique SAXS for small angle X-ray scattering. So this is the basic setup of the experiment. We have the X-rays coming in, our capillary containing a small volume, maybe 10 to 20 microliters of sample, and it's just an aqueous solution typically. And the X-rays scatter from a well-collimated beam onto this 2D area detector, and we can perform a azimuthal integration in order to get a one-dimensional scattering profile. And this is the final sort of raw data that we get from the experiment, which is intensity as a function of Q, where Q is what we call the momentum transfer, which is effectively the angle of scattering on the detector. Now, ideally, these are monodispersed samples. So what I'm going to talk to you about today assumes that the samples are monodispersed. There are different techniques that you can use mathematically to try and deconvolute if you have polydispersed mixtures. But today we're just going to talk about monodispersed samples. But if we can analyze this one-dimensional profile, we can get a lot of information about the overall size and shape of these particles. So this is sort of a quick representation of lots of different scattering profiles overlaid on one another so that we can see the types of signals that we get. At low Q values, at small Q values here, we're looking at the SAXS region, the small angle scattering region. We're getting the overall size and shape of the particle. This is where we get more photons. This is typically where we get enough signal to noise for these experiments to be doable. If we could collect much higher quality data, which you can with special arrangements of your instrumentation geometry and very high concentrations of protein, then you can get information about the overall fold and even the atomic structure, though this is very rarely done in practice. Most of the time, we're looking at just small angle regions.
Tom, could I ask on the
Yes.
previous slide, the variable Q, so
Mhm.
it came from the angle, right?
Yes. So it's the sine of the angle on the detector here. So we have 2 theta is the angle of scattering, and Q is defined as 4 pi sine theta over lambda, where lambda is the wavelength of the light. So if we have a 1 angstrom wavelength, our Q values are typically for a small angle experiment somewhere between 0 and 0.5. That's typically the range of data that we're collecting. And that effectively is just where the edge of the detector is, right? So if we go back, we have a square detector typically. It's a radially symmetric image. So here at the low Q, we're looking at the center of the detector as a Q of 0. And as we go to higher and higher and higher angles on the detector, we're getting higher and higher and higher Q values, where the corner is maybe 0.5, 0.6, 0.7, something like that.
Okay, so now I have understood why it's 1 over angstrom.
Yes, that's right.
Because
It's 1 over angstrom because it's 4 pi sine theta over lambda, and lambda is in angstroms.
yeah, thank you.
All right, so what can we get from a SAXS experiment? We can get things like the radius of gyration, which is kind of a descriptor of the overall size of the particle. It's like, kind of think of it like the radius of a sphere, but particles are not all spheres. And so this accounts for the shape of the particle as well. The maximum particle dimension is exactly what it sounds like. It's just the maximum length, the largest vector that is in the particle. The molecular weight is the mass of the particle, so we can get that from SAXS experiments. And we can also get what's called a oligomeric state, which means if we have 2 proteins that come together and form a complex, we can figure out how many particles are in the complex. It could be 2 of them, it could be 3 of them, it could be 4 of them in order to get the oligomeric state. In addition to that, we can also get how they're arranged in space. It's not just the number of them, but how many are arranged in a particular configuration. Something that over the last 10 to 20 years has become very popular is the study of flexible regions of proteins. This is really important for understanding a lot of different diseases because a lot of proteins are intrinsically disordered. They don't actually have a single rigid structure. They actually adopt many different conformations and have some regions that are unstructured entirely until they interact with another protein, for example. And understanding that helps us understand intrinsically disordered regions that are parts of diseases for things like Alzheimer's and a lot of neurological disorders, as well as just understanding the concepts of the folding and unfolding pathways of proteins and how they actually fold. So SAXS is useful for a lot of those different types of experiments. And with some advanced modeling techniques, we can get things like low resolution molecular envelopes, the overall shape in 3D, as well as actual atomic models representing the data. So what I want to talk about mostly today is something called the pair distance distribution function. Okay, so the pair distribution function is kind of like the primary real space representation of this information content that's in the scattering profile. And that's because they're related by a Fourier transform. So if you take the Fourier transform of I of Q, you get what's called the P of R, the pair distribution function, which is a real space representation. And we can see down here that these are related through a sign transform. Okay. So what is the P of R? So the P of R is a histogram of all of the interatomic pair distances in the particle. Okay, and this is really important because this tells us about the shape of the particle. Okay, so we have some example P of R curves down here, some really simplified ones. So if we take something that's like a sphere, you get this green P of R curve, which is kind of like a Gaussian bell curve in a way, right? If you look at this orange cylinder, which is sort of a long rod, you get a lot of short distances at small r values, so a lot of short distances. And then you get fewer and fewer and fewer long distances. Okay, so this sort of right skewed P of R curve tells us that it's an elongated object. If
Tom,
we look, for example, yeah, go ahead.
could I ask about this histogram definition? Um,
Sure.
uh, Usually, a histogram is a discrete object, so a finite collection of distances, right? And here, indeed, a protein is a bounded object with finitely many atoms. But you show continuous curves, right? So could
Yeah,
you clarify?
Yeah, so mostly this is because we're looking at low resolution. So if you think about an atom, yes, you could, if you took an atomic model of a protein and you looked at the centers of all the atoms and you calculated the distances between them and then calculated a P of R curve, yes, you'd get a sort of jagged, noisy, discrete histogram with bins. But in reality, the electrons are not centered at the center of the atom, right? They're this diffuse cloud. And because they're this diffuse cloud, we're actually looking at all the inter-electron pairs, technically, rather than the interatomic pairs. We're actually looking at what the X-rays scatter off of, which is the electrons, not the centers of the atoms. And so we get a smoother function. But it's the same basic thing. It's really just how finely you're sampling this curve. So we get in practice, it is a histogram, but we view it as a curve simply because of the number of bins that we have in a way. Even if you had an infinite number of bins, it would still look smooth simply because, again, the particle is an electron cloud, which is, electrons don't have a single position. They're this sort of diffuse cloud around the center of the atom.
Okay,
So
thank
if we
you.
look at this dumbbell, this red sort of dumbbell looking shape, you can see that we have two peaks. And these two peaks come from the different types or the different length vectors that are present in the particle. So at low r values, we see this large peak, and that comes from vectors within each of the individual domains. So we have short distances within the domains. But then we have the second peak at long r, which is the vectors binned from the vectors between the domains, right? And so we get this sort of bilobed distribution. So this P of R curve is really the key physical descriptor of the particle structure in one-dimensional real space. So getting this P of R curve is really important. So in order to calculate this P of R curve, we use a Fourier transform, right? And so I gave you this equation before, which is a sign transform of the intensity function to get this P of R curve. And this is what happens when you take real experimental data and you get this P of R curve. You get this really wiggly, ripply, artifact-filled P of R curve, right? And this is useful, but it's not terribly useful for figuring out what the real P of R curve is of the particle because the actual particle doesn't have all these ripples in the P of R curve. The reason that this happens is because our equation here is an integration from 0 to infinity. So it's over all Q space. But you notice that our detector has an edge, and therefore our Q range has an edge. We don't have an infinitely large detector, so we have to cut off the Q range at some value. What Q value that is depends on the geometry of your experiment, how far your detector is away from the sample X-ray interaction region, and how large your detector is. But the other thing is that the data have noise in it, right? These are, of course, real data, and so they have experimental noise. And so when you take the Fourier transform of this experimental, noisy, finite curve, you get this flawed P of R curve, right? And so people have tried to come up with some different techniques to solve this problem. And the primary one is something called an indirect Fourier transform. And the basic idea behind an indirect Fourier transform is that it focuses on the P of R curve itself. It says we know certain properties about the P of R curve. We want to restrain the P of R curve to be a smooth continuous function. And so we want to parameterize the real space P of R curve with a set of basis functions that will allow us to do that. And because they're related by this Fourier transform, what we can get from the P of R curve with these basis functions, we can get through the scattering profile itself by going back to reciprocal space and fitting that scattering profile, that experimental data. This idea of indirect Fourier transforms was pioneered by Glatter in the late 70s, along with Peter Moore. And then over the next 10 or 15 years, into the 90s even, it was developed by people like Svergan and Henderson and Peterson in order to add restraints and try and improve our ability to get information out of these scattering profiles. Most of what we're going to talk about today is Moore's approach, and I'll explain why in just a little bit. But Moore's approach was to show how scattering profiles can be expressed using Shannon information theory, and we'll get into the details of that in just a little bit. But before we do, let's talk about the sort of standard indirect Fourier transforms that most people use. So this is the original transform defined by Glatter, and then was largely popularized by a software program called GNOME, which is out of the SAXS suite of SAXS analysis programs. So if you're familiar with SAXS, it's the largest software platform for BioSAX. And GNOME is their program for calculating P of R curves from scattering profiles. And the reason why it became so popular was because it implemented Glatter's approach with some new regularization terms and allowed you to have a nice, convenient software package to be able to do this easily. So these algorithms use cubic splines as the basis set to model and parameterize the P of R curve. And so if you're not familiar, cubic splines are effectively a set of polynomials that you stitch together in a piecewise manner through these sections defined by what are called knots. These knots are like hinges of these piecewise regions. So you multiply the cubic spline by like a Heaviside step function, and that gives you this sort of top hat region where the spline is, active in a way. I'm only showing 5 knots here, but in reality, typical experiments or typical analysis will use maybe 20 to 40 knots, depending on the size of the particle. And that's a key thing, is that the maximum dimension is something that restrains the number of knots, right? And we'll talk about the maximum dimension in more detail in a minute, but it's a really important component of all of this analysis that we're about to get into. Using cubic splines, you can see that anything piecewise is not really what would you say? It's not really good mathematically, right? You don't necessarily like piecewise functions. They're not closed form analytical solutions. And that means that when we need to get size parameters, things like the radius of gyration or the particle mass or the volume, we have to do things like numerical integration and optimization. And that's how we get these cubic splines, is through numerical integration. So it's all approximations because we're using these cubic splines. So I want to talk about Dmax for a moment. So this is where Moore's approach came to bear in the field. So Moore tried to apply information theory, the concepts of sort of communication, to solution scattering. And the basic premise of information theory, Shannon's information theory, is that an exact continuous signal can be recovered from the discrete sampling of that signal, provided that the sampling rate is at least 2 times the highest frequency present in the signal. This was the fundamental proof of information theory by Claude Shannon back in the 40s. So I want to go back to this image here of the experimental data. So you can see that we have a noisy experimental profile. But underneath this profile, you can see that there's a nice, smooth, continuous function. And that continuous function is the scattering profile that we are after. We want that continuous scattering profile so that we can get a good P of R curve and get good size parameters and structural information rather than this noisy scattering profile. So in the context of Shannon theory, that continuous signal can be recovered from that discrete sampling, right? Our experiment is effectively a discrete sampling of that underlying continuous scattering profile, provided that the sampling rate, in other words, the distance between the adjacent data points, is at least 2 times the highest frequency present in the signal. And in SAXS, the highest frequency is related to the maximum dimension of the particle, Dmax. So in SAXS, if we have q values that are 4 pi sine theta over lambda, we can relate the vector length to a frequency in reciprocal space. And that's 2 pi over that vector length in the q values that we're using. So that means that the corresponding frequency of Dmax is 2 pi over Dmax. But Shannon theory says that we need twice the highest sampling rate. And so that means that we need to make sure we're sampling the data at least 2 pi divided by 2 Dmax, which is effectively pi over D. Okay, so that means that we need to sample the data at a width in Q space that is less than this channel width of pi over D. And so that gives us an estimate for the number of unique pieces of information in a scattering profile, or the number of Shannon channels in SAXS data. And that's our total Q range divided by the width of that Shannon channel. So that's Q max divided by pi over D. And in practice, we end up with a number of Shannon channels typically around 10 to 30. And so what that means is that in a typical experiment, we only have around 10 to 30 independent pieces of information from which we get all of our structural information from. So this is a fundamental property of SAXS experiments, is that they are low information content. Okay, they don't give you high resolution structures. They only have a certain amount of information, only 10 to 30 unique pieces of information. So Moore wanted to get this information. He wanted to extract those independent pieces of information using Shannon information theory. And his real contribution to this was recognizing that SAXS is essentially a band-limited signal. And so he wanted to get at this information, and in order to do so, he recognized that I of Q and P of R are Fourier transforms of each other. So he wanted to find a set of basis functions rather than cubic splines that were instead Fourier mates of each other. And in order to do that, he came up with these two auxiliary functions. One is called U of Q, which is Q times the intensity, and the other is called Q of R, which is the P of R curve divided by R. And the main insight here was that because the P of R curve is bounded as a band-limited signal, it has a maximum dimension of D, he can represent Q of R, which is also thus bounded, by a sine series, a Fourier sine series. And so this was the real innovation of Moore's approach, is that rather than representing the P of R curve using cubic splines as the basis set, he's using a sine series as the basis set. And the advantage of this is now U of Q and Q of R are Fourier mates. An advantage of the sine series also is that this is an orthogonal basis set, which means that we have independent information from each one of these terms in the series. All right, so we don't typically use Q of R and U of Q directly. We use P of R and I of Q. So we can do that calculation and show that the P of R function is this sine series, but modified. We have an extra R term here. And similarly, we have the I of Q set of basis functions, which is this equation here. So we've shifted the equations from being continuous integrals to these discrete sums, which allows us to solve them with tools like least squares, which we'll get into in just a minute. But the important thing to note is that because they are Fourier mates, we have the same coefficients, these ANs, which define the weights of the basis functions, both in reciprocal space and in Fourier space, or in real space. So if we can figure out what these coefficients are, we know what our equations are in both domains. So we can use least squares in order to determine what these ANs are. We can set up a system of linear equations using the data set and actually fit this in order to determine what these AN coefficients are. And once we do that using our scattering profile, we can now use those ANs to calculate the P of R curve. And since we're using least squares, we now have a full closed form analytical solution to this problem, and we can get rigorous error propagation. That was really one of the primary reasons why Moore developed this, was because the cubic spline approach doesn't allow you to get rigorous errors and uncertainties on the data points and on the coefficients and on the size parameters that we're going to be extracting from this. And so using least squares allows us to get rigorous error propagation because we have the variance and covariance matrices. And that allows us to get uncertainties on the coefficients as well as the size parameters. But this approach has some problems. One is that these are some abstract coefficients. They don't have any physical meaning to them, right? They're just the weights of these basis functions, so they're not physically meaningful. Another is that, which is a more practical problem, is that it was shown in the decades following Peter Moore's publication, was that his method was a little bit more susceptible to ripples and instability than the cubic splines because the cubic splines had regularization that could be applied to them. Nobody had figured out the regularization problem to restrain the smoothness of the P of R curve from Moore's approach. So the reason for this was that Moore wasn't really trying to solve the smoothness problem of the P of R curve. He was really just attempting to show how information theory could be applied to scattering and get rigorous error estimates from those parameters that we get from them. So in practice, his method ended up not being as popular. There wasn't a nice piece of software that people could use like there was with GNOME. So GNOME was the one that really became popular. So this was where I came in to the sort of the field was maybe 10 years ago, I started learning about this and I decided that I wanted to come up with my own tool to solve this problem. And so the first step for me was to come up with a different set of basis functions that allow me to have coefficients for these basis functions that are meaningful values, right? So one of the things that you can gain from Moore's paper was that there are some special Q values because we're setting up a series, a sine series. And these special Q values are kind of like these nodes of this series. So if we look back at the sine series, you can see that we have n pi r over d. So we kind of have a fundamental frequency d. And these nodes can be related to these Shannon points that we talked about, where the Q values are n pi over d. So the Shannon points, So the Shannon points define what I call Shannon intensities. Okay, so these are physically meaningful values. They're effectively just the intensities at the Shannon points. But we don't have Shannon points in Moore's equations, so I went ahead and derived a relationship between the Moore coefficients and these Shannon intensities. And it turns out it's a fairly straightforward relationship, so I can then go and rederive all those equations. And now I have a new set of basis functions, so I effectively have performed a change of basis, where I have the Shannon intensities are the weights for these new basis functions. So these are now physically meaningful coefficients rather than just abstract weighting coefficients. These are the new basis functions. You can see that they are a sinc term basis function, and we can see them over in the upper right here. Okay, so the basis functions are dependent on the size of the particle d. And so for a particle with size of d equals 50 angstroms, we get this series of basis functions for just the first 8. Now let's look, for example, at basis function 7. Okay, it's this pink curve. So it starts at a value of 1, and you can see it decays like a sinc wave, right? And eventually, when we get over to 7 pi over d for the 7th basis function, which is sort of this 7th harmonic mode of the Q values, we can see that this basis function has this large peak and now dominates the intensities at that region. And so you can see that where these n pi over d nodes are, we get all of the other terms to cancel out. And this is just a property of these basis functions being orthogonal to one another. So we're extracting this independent information from these terms. So we can sort of visualize this real quick here. We have an experimental curve on the left, and we can look at the intensities at the Shannon points as these blue circles. And if we extract those intensities, then we can multiply them by the basis functions, sum them all up, and we get the overall red curve that you're seeing. So we now have a functional, continuous form of that underlying scattering profile. But of course, we don't really want to use values right at those Shannon intensities and just extract the number. For one, we don't usually measure the intensity exactly at the Shannon point, right? And two, we have noise. So if we only did that, then we'd have to interpolate between values that are noisy, and we'd get a really rough estimate. And we're missing out on all this other data that we have, right? And so because of how we've defined these equations, we can set up a system of linear equations using least squares and solve it algebraically. So here we have what's called chi-squared. Chi-squared is our overall goodness of fit metric. And so effectively we have our a sum over all of the individual data points. Maybe there's 500 data points. We have an error bar or an uncertainty on each data point, and we do the residuals squared. So we have the experimental data minus the model for the data at that Q value. And so we can take this equation and we can take the derivative of it because we want to minimize it. So we take the derivative of it and we set it equal to 0. And we end up with a system of linear equations that we can solve algebraically. On the left-hand side, we set that equal to a vector m, ym. And on the right-hand side, we set that equal to a matrix Cmn, and we solve for these in coefficients. So we end up with a system of linear equations that we can just use some linear algebra to solve for the inverse of C. All right, and so this allows us to utilize this extreme oversampling of the experimental information, right? So we have maybe 10 or 20 or 30 unique pieces of information, unique Shannon channels in the data, but we have 500 data points, maybe 1,000 data points. So we have something like 50 unique measurements for every one of those pieces of information. And so by using the entire scattering profile, we get a much more precise estimate of what these coefficients are. Also, because we have this C inverse matrix, that is the covariance matrix from least squares. So that allows us to have some rigorous error propagation for the uncertainty estimates on those coefficients and ultimately for the size parameters that we can derive from them. But we still have fundamentally the same problem that Moore had, which is that the Moore approach and subsequently our approach still has this sensitivity to Fourier truncation ripples because we don't have any regularization. Now, other techniques like Noam and Glatter's technique that I mentioned, these have been popular because they have regularization techniques applied. In this case, we're looking at Tikhonov regularization of these indirect Fourier transforms. The idea is that we know that a P of R curve should typically be a smooth function, especially at low resolution, the low resolution of SAXS. The P of R curve should be a smooth function. It shouldn't have all these ripples in it. And we can use that as a restraint to regularize the coefficients. So we can monitor the smoothness of the P of R curve using the second derivative. The second derivative effectively looks at the curvature of this curve and tells us kind of how wiggly it is, right? How many oscillations are in there and the severity of those oscillations. So we define a term S, which is the integral over the second derivative squared of the P of R curve, and that tells us a number estimating how smooth the P of R curve is. So rather than only minimizing chi-squared, which is the goodness of fit in reciprocal space, we also add this smoothness parameter, which is how smooth the real space representation is. So now we have reciprocal space plus real space, and this defines some kind of scoring function. However, because S is not on the same scale as chi-squared, we need a Lagrange multiplier to put them in the right weighting. And alpha is a user-defined weight that says how smooth do you want the data. So if you make alpha 0, then you're back at your original starting point of just chi-squared. So there's no smoothness considered, and it's just looking at minimizing chi-squared. But if we have a large alpha, then we can over-smooth the P of R curve, and it might not fit the data particularly well. So finding a good balance of alpha is an important part of this program. Now, previous methods like Noam are all numerical, as I mentioned, because of these cubic splines. They're not analytical. And so they're based on some subjective interpretations. They're looking at differences in the knots and things like that. And so they develop some perceptual criteria because of this numerical optimization. So I wanted to set out to define this regularization for our method using Shannon intensities. And an analytical solution to the regularization problem doesn't exist currently, or at least it didn't until we figured this out. So we applied Tikhonov regularization again with the second derivative of P of R. So if we look at the second derivative of P of R, our P of R function, we get this equation. But if you look at S, we can't actually square and integrate this, right? Because we have this infinite sum. So that means we can't solve this. This is one of the major reasons why people never applied regularization to Peter Moore's approach because the whole point of his approach was having a closed form analytical solution, and then applying a numerical solution would just sort of make it pointless. It would break that nice mathematical behavior. So the trick that I realized was that we don't actually need to solve t directly. What we need is to minimize t, right? Because we just want to use t, this scoring function, to figure out what the optimal coefficients are, these INs. And if we only need to minimize t, ultimately that means we just need to take the derivative of t with respect to the coefficients. So we don't need s explicitly because of that. We just need the derivative of s with respect to im. And this allows us to get around this problem of not being able to solve this integral that we saw in the last slide. So if we take the derivative of s with respect to the coefficients, we can bring that differentiation inside the integral. And we can actually take the derivative first with respect to im rather than trying to integrate first. And if we take the derivative first, here's our big second derivative of p of r equation. We can now bring down the square and expand all the terms and then exchange this infinite sum with the integral because of the Fubini theorem. And that allows us to then expand this. And there's a ton of terms, and it took a while to figure out, but eventually we can get all of these integrals to converge to 4 large terms and combine them all, and we end up with this matrix. Okay, so we have a sum over all of the Shannon channels. The INs are the Shannon intensities, and this GMN is this large matrix. Okay, so we now have a closed form solution of the derivative of s with respect to the coefficients. We can go ahead and plug that back into our t equation for minimizing t. So we need the derivative of t with respect to im. We've already solved this derivative of chi squared earlier that I mentioned, and now we're adding in this term here. And so we can do that the same way that we did before. Because of the nice properties of these equations, we can actually pull out the Shannon intensities from both the s term as well as the chi squared term. And we end up again with a nice system of linear equations that we can solve for, okay, algebraically. And so you can see that we have this alpha GMN term. Again, a user has to define what alpha is. It's an input, it's not an output. And so we have to tell it what alpha is in order to tell it how smooth do you want your P of R curve to be. In other words, this algorithm is now figuring out what are the optimal Shannon intensities to best fit the data and have a smooth P of R curve given the weighting that you are telling me to give it. Okay, so we can apply this. We developed a bunch of code for this, and I'll show you actually an example of this in just a few minutes. But here on the left, we have the standard approach of our algorithm without regularization, or equivalently with setting alpha to 0. And then once you set alpha to a good value, you get a nice smooth P of R curve. But importantly, we get not only a smooth P of R curve, we also get a good fit to the scattering profile. And that's really the key thing, is figuring out what that alpha is. All right, and once again, we have rigorous error propagation, just like we had previously, because we still have this covariance matrix C. Now the covariance matrix includes that regularization that we just figured out. And so we can get the error bars, the uncertainties on the coefficients themselves, but also because we have the functional form for the scattering profile, for the continuous scattering profile, and for the P of R curve, we can calculate them at any Q values and any R values that we want, and therefore we can get the uncertainties on each one of those calculated points. All right, now that was all sort of the basic functional form of these equations, but really this is kind of the cool math that I wanted to eventually get at. So I'm going to wrap this up with talking about the parameters that we can get from this new closed form solution of SAXS, right? That's effectively what we've done, is we've come up with a closed form solution of scattering. And this is really the key point to take away from all of this, is that the Shannon intensities, the INs, represent the complete band-limited information content of the experiment. And that means that whatever information you could get from either the I of Q curve or the P of R curve, you can also get, in theory, from the INs directly, because they encapsulate all of this information. So we've reduced something that was previously hundreds and hundreds of data points to something that is now 10 or 15 unique numbers, these Shannon intensities, right? So all of the information is embedded with within this discrete set of intensities. So this allows for some really nice math. So let's take a quick example, something called the forward scattering, where we're looking at Q equal to 0, okay? So this tells us information about the mass of the particle, and it's kind of an overall normalization constant for normalizing all the various aspects of our experiments, like the beam flux and the sample thickness and the sample concentration. They all kind of get absorbed into this forward scattering as a normalization constant. And so we often need this value very accurately to do things like estimate the mass of the particle. So we can go through this equation. We can say, okay, if this is our equation defining I of Q based on the INs, what happens if we just plug in Q equal to 0? Okay, so Q equals 0 here. We plug in here, so we get 0 here, and we now get this whole fraction going to 1. And this term is a sinc function. So sinc of 0 is 1. So this whole thing goes to 1. And so we actually get this really nice, simple, elegant equation for the forward scattering directly from the INs. And this was something that I don't think I've certainly never seen this. I never knew this before I saw this. I don't think anybody did. And so this is a really nice equation. And the reason why this is important in practice is that we don't measure I of 0 because Q equal to 0 is behind the beam stop. The X-rays, the primary X-ray beam, it hits a beam stop and blocks the very, very low Q region. So we only typically get down to Q values of around 0.01 or so. So we don't get anything behind the beam stop. So we can't measure the forward scattering. So we have to extrapolate. So usually we take the first 20 data points and we extrapolate to what I of 0 actually is. And now we actually have mathematically a true measurement of I of 0. Now this is a sum to infinity, and like I said, we don't get an infinite number of Shannon channels, of course. But because of the way a scattering profile works, most of the photons, most of the X-rays, and most of the intensity are at the low angles, right? Most of the intensity is within the first 4 or 5 Shannon channels. So this is a really neat, mathematical result that we can look at what is the forward scattering I of 0, and we can say, okay, it's Shannon channel 1 minus Shannon channel 2 minus Shannon intensity 3 plus Shannon intensity 4, and we go back and forth, right? So it's 1 minus 2 plus 3 minus 4, and this is on a log scale, and so most of that is within the first 4 or 5 Shannon channels, and the rest of it is just sort of oscillating around a particular converged value. So we get a very, very accurate estimate of I of 0 just from these Shannon intensities.
Tom, could I ask about this values where I of Q, is it really 0 or is it very close to 0?
You never measure Q equal to 0 because you
No,
have this beam
sorry,
stop.
I of Q. So when the graph approaches the horizontal axis.
No, it's never 0. It theoretically could be 0 because there are certain, like right here where we have this is on a log scale, keep in mind. So it's sort of going down and it's just barely touching. Yes, theoretically at this point, it is 0. You will pretty much never measure exactly that point, right? It's an asymptote in this log representation. But yes, theoretically it does go to 0. Everything cancels out. Like if you look at a sphere, it will cancel out at certain values. It's just with noise of the experiment, you'll never measure 0. You might actually measure negative numbers just because of the noise in the experiment.
These values of Q, are they also, can they be written analytically?
Can the values of Q be written analytically? You're talking about where these nodes are?
Yes.
So this particular curve is for a sphere. And so, yes, you can figure out what these nodes are for a sphere, but most scattering profiles don't have these nodes. This is just because it happens to be a sphere, and spheres happen to be nice mathematical treatments that do cancel out exactly at these locations. But most scattering profiles don't because they're weird shapes of proteins.
Okay, so it's a special case.
Yeah. All right. So I of 0 is very easy to derive, but another parameter that we have is the radius of gyration. This is not easy to derive. It turns out it was quite complicated, but we could do it because we have, again, this closed form solution. So this numerator is r squared times p of r. We can plug in our p of r curve there. It's effectively the second moment of the p of r curve. And we can plug our p of r curve there with our INs, go through several rounds of integration by parts and solve this. And we end up with this equation for the radius of gyration. If you're more interested in all these mathematical details, you can go to the supplementary material of the paper that's cited throughout here. And so we can get radius of gyration directly from the INs. This is something, getting these parameters directly from the Shannon intensities is not something you could have done previously, right? That's something that's exclusive to Moore's approach because he has this full closed form analytical solution, which we've built on. All right, so you can't do this with something like GNOME. GNOME has to get the radius of gyration by actually numerically approximating these integrals using things like the trapezoid rule. And we can do the same thing with volume. Volume is another common parameter. This is something, this Q value, this Q here, which is not to be confused with the other two Qs, which the lowercase q is the x-axis. The other Q that we had earlier was this more Q of R thing. This is a different Q called the Perot invariant. Don't ask me why all these scattering theorists decided to use Q for everything. But this is called the Perot invariant. And the Perot invariant is this integral of q squared times I of q. And we can, again, plug in our formal function, our continuous functional definition of I of q, and solve it through all these rounds of integration by parts. And again, we get q is equal to this sum as a function of the INs, okay? And now we can plug that into this equation for the volume, which is a fairly simple equation, which allows us to estimate the volume of a particle directly from the INs. So I want to give you just a couple examples with a solid sphere, which I think really showcases the power of the math. And just as a mathematician was really cool to see. I just really love when math is sort of like, just converges to these really nice examples. So the sphere is one of the few shapes that have an exact definition for a scattering profile that have been derived, okay? This is the equation where r is the radius of the solid sphere. So what we can do is we can plug in, we can say, I want to know what the Shannon intensities are by saying Qn is equal to n pi over d, where d is equal to 2r. And so I can plug those in here, and I can work all that math out, and it turns out that it converges to this equation here. And the key thing I want you to recognize about this equation is that r is not present anywhere in this equation, and that is on purpose. And the reason for that is because what we've done with our orthogonal basis set is we have separated and decoupled the concept of shape from the concept of size, right? Where size is defined by that fundamental frequency of the basis functions d, and the shape is the INs. And we can see that in this example where we have the analytical expression for a sphere, that there is no r anywhere in this equation. And what that means is that if we were to actually have spheres of different sizes and look at their scattering profiles, say we've got a radius of 15, a sphere with a radius of 25, and a sphere with a radius of 50, we can see what these scattering profiles are. You can see that 15, it's kind of just like dilating the x-axis almost, right? And so the Shannon intensities themselves are the same values. The y-axis is the same value for all of these different spheres. It's not dependent on the size of the particle. The Shannon intensity themselves are the same. It's only their locations, which are defined by the QNs. It's just the fact that QN now has n pi over d, and d is larger or smaller, and it scales that value. So we've successfully decoupled the concept of shape from the concept of size. So you can see here an example of this in practice. We have the noisy scattering profile in the gray dots, and then we can fit that with our least squares equation, and that gives us the IN values in the blue circles. And then we can reconstruct the scattering profile from that at those Q values, and you can see the red curve. And then we can compare that to the actual ground truth, which is the equation of the sphere I showed you in the last slide. And you notice that the red curve is faithfully reconstructing, even though the data are that noisy, is faithfully reconstructing that underlying scattering profile of the sphere with far more precision than you can get just from that noisy data. And we can look at the same thing for the P of R curve. We get a very nice representation of the P of R curve. All right, and finally, I just want to highlight some really nice aspects of this math that I mentioned for the sphere. So we can take those equations that we had, we can plug in, this is the equation that we derive for RG, right? We can plug in the IN values for the sphere, and we end up with this large equation, okay? It's this large summation. This is a pretty ugly equation, especially when you actually expand out all the terms. But it turns out that you can actually take this entire thing and every single one of those terms converges when you combine them in the right order. And we can actually take this entire summation, believe it or not, it all converges to the fraction 3/20ths. This entire crazy summation all converges to the fraction 3/20ths. So we can plug that in and we get the radius of gyration of the sphere is 3d squared over 20, and d is just 2r. So we can plug in 2r, and that means that rg squared is equal to 3/5 times the radius squared. And this is, in fact, the well-known identity from geometry for the radius of gyration of a sphere. It's just, in my mind, when I saw this, I was like, what a beautiful mathematical result to see us being able to get the radius of gyration of a sphere through this really complicated method using just scattering intensities. So it's very neat. We can do the same thing with the volume of the sphere. I told you we can measure the Perot volume using this Perot invariant. We can have this equation for the Perot invariant, plug in the IN values for the sphere, and we can get this value for the Perot invariant of the sphere, plug that into the Perot volume equation, and we get an answer of 4/3 pi r cubed, which is, of course, the volume of a sphere. Again, a really neat equation or result. We've implemented this all in software. We've published this open source a few years ago. So Dense is a package that I published a few years ago, which allows us to do 3D reconstructions of electron density from solution scattering. And we've taken this whole pipeline and built it into Dense because we need these smooth curves for Dense to work. So this is now part of Dense. It's also implemented in a popular package called BioXS Raw, if you happen to do this stuff. And so this is a quick example of our little GUI. We made a little GUI for it. And this GUI has automatic estimation of Dmax. So it'll give you a quick estimation. It'll run the analysis and give you an estimation of what Dmax is. And then it'll actually also estimate for you the optimal alpha. So a good balance. And there's a lot that goes into those steps. So our GUI here has some interactive sliders, and we calculate a slew of maybe half a dozen or so size parameters that you can save. So I thought what I could do is a quick demo of this just to show you how it works. So I've loaded it up here. Hopefully you can all see that. I'll make this a little bit bigger. So what we have here is the raw scattering profile is in the black dots. And when we load this, it automatically estimates a Dmax of 126 angstroms and an alpha value of 71. That's what it estimated. But as a user, you can say, okay, I think the P of R curve should be a little bit smoother. I think the Dmax should be a little bit larger, right? And so the reason why I wanted to pull this up is because you can see the effects of what happens when you change these numbers, okay? Now, I'm going to expand this a little bit. You can see the P of R curve is nice and smooth. If I make the Dmax longer, right, it effectively just stays flat, right? It just goes to 0. It doesn't matter what Dmax I use because there's no more vectors. This is actually extracted from the data. There's no longer vectors, so it kind of just stays flat down at 0. What you notice is if I take this and I make it too short, what happens is now the curve, there's not enough frequencies in the curve, high frequencies, and so it can't fit very well. So it breaks, right? Because it doesn't have the ability to fit these features because these features come from the large vectors in the particle. And so there's this signal in there that's higher frequency. So we have to have a large enough Dmax to match that frequency. And this is one of the ways that we know what Dmax is, as we see at what Dmax do I get a good chi-squared. You notice that I have chi-squared measured up here, right? So I have a high chi-squared when Dmax is too small. Chi-squared should ideally be 1. So I can see where it goes to 1, and it's right around there. So that's how we estimate Dmax. We can also look at the P of R curve and just see where does this curve naturally fall to 0 if I give it a really large number, right? The Dmax isn't actually 200. I can just look at the P of R curve and say, okay, this naturally falls to 0 around 126, right? Something like that. Similarly, I can play with the alpha value. If I set alpha to 0, I get a few more ripples in my P of R curve. Okay, and so this is the standard Moore calculation, right? Or our modified calculation. With no alpha, you get a lot of ripples. But if I reset the values, I can get a nice smooth P of R curve and still have a good fit to the data. Now, if I make alpha too large, you notice that I get a smoother and smoother, if I do it sort of extreme, I get a smoother and smoother P of R curve, but now the fit to the data gets worse and worse, right? So as a user, I have to balance a good amount of smoothness with a good fit to the data. And for our purposes, we always err on the side of a better fit to the data. So we have something that allows us to get a nice fit to the data, and then we just go at 10% above the chi-squared. So it's a fairly quick way for automating this. And then we get all these size parameters along with their error bars at the bottom there. All right. With that, I will wrap it up. And then, oh yeah, I just mentioned that we use this indents for generating these three-dimensional electron density maps. So there's a lot of practical utility for getting these curves accurate. All right, and finally, I'd just like to acknowledge my group who have helped a lot. A lot of them have joined since I did all of this, but we all use this and develop it and develop our dense package together. I'm funded by NIH and NSF, and I've had a lot of help, people reviewing the software and the math for me. So I'd like to acknowledge all of their hard work as well. And with that, I'm happy to take any questions.
Thank you, Tom. Please thank Tom for a nice presentation. So, physically or virtually. Thank you very much. I'll stop the recording.